How does it look? Sanford. Good evening, everyone. Bismillah ar-Rahman ar-Rahim. You hear me? Everyone hears? Can we start? Bismillah ar-Rahman ar-Rahim. Good evening, everyone. It's my great pleasure and honor to be chairman of this session. We have a distinguished speaker this evening. The Sheikh Hamdan bin Rashid Al-Maktoum Award for Medical Sciences was established in 1999 by the visionary of the late Sheikh Hamdan bin Rashid Al-Maktoum. His Royal Highness has been driving force to support the medical sector. This prestigious award will live on to be a source of inspiration and motivation for researchers around the world to have a great positive impact on the humanity. His Royal Highness will always remain in our hearts. The award collaborated for decades with academia and healthcare institute to fund distinctive research and honor excellence, in which has a great impact on the humanity. These efforts have paid off as two of the Grand Hamdan Award winners were awarded the Nobel Prize in Physiology in the 2010 and 2020. The winners were Sir Robert Edwards and Dr. Harvey Alter. In addition, I would like to welcome the Board of Trustees members, whom are great supporters of the award virtual activities. Now it's my honor and pleasure to present an eminent pleasure to introduce Professor Sanford Marquez, the winner of Hamdan Award for Medical Research Excellence in 2015, for his remarkable contributions to colon cancer research. Professor Marquez is a medical oncologist and cancer researcher. He obtained his undergraduate from Harvard College, then pursued his MD degree PhD at Yale School of Medicine and University of Chicago. Professor Marquez completed the fellowship at National Cancer Institute, and currently he's appointed at Case Western Research University and the Case Comprehensive Cancer Center. If I want to go on and on, I think I will need more time than the time allocated for the lecture to talk about our distinguished speaker tonight. So without further ado, I would like to kindly ask Professor Marquez to begin his presentation titled Biomarker-Based Non-Endoscopic Detection of Barrett's Esophagus for the Prevention of Esophageal Carcinoma. Professor Marquez, please. Professor Marquez, can you hear me?
Yes, can you hear me, Dr. Abu-Hamor?
Yes, sir. Well, kindly, you can present. You can start your presentation.
Well, let me begin by thanking Dr. Abu-Hamor and my great friends at the Hamdan Awards for the pleasure, the privilege of being able to give this address, which is, of course, dedicated to the memory of the late Sheikh Hamdan, who founded the awards. Let me begin sharing my screen. Let's see, can you see my screen in presentation mode? Good, okay. So I'm gonna just, there we go, okay. So the disease I'm going to talk about today is how to approach esophageal adenocarcinoma through non-endoscopic detection of Barrett's esophagus, the precursor lesion of that disease. All right, let's see. And I know there is great interest in the medical community today in blood tests for cancer, but what I'm going to show you is that this is an instance in which blood tests are not the answer. These are my disclosures relating to technology that has been licensed by Exact Science and Lucid Diagnostics from my lab. So this story actually begins many, many years ago when I had just completed my oncology fellowship and met this patient, a 33-year-old accountant in Cleveland who had metastatic colon cancer. And more remarkably, so had his father and so had his grandfather. And this was the first time I was seeing a family that we now recognize as having Lynch syndrome. And together with our collaborators in the Vogelstein laboratory, we were able to participate in showing that inherited colorectal cancer Lynch syndrome was caused by mutations in DNA mismatch repair genes. But one of the initial surprises in those studies was that mismatch repair deficiency was not seemingly inherited in all of the individuals in these families. And let me just go forward. And so the question became, what was the cause of mismatch repair deficiency in individuals who did not have a family history of colorectal cancer? And ours was one of the laboratories that contemporaneously with several others showed that the basis of sporadic deficiency of mismatch repair capability in colorectal cancer was epigenetic silencing of DNA mismatch repair genes, particularly the MLH1 gene, so that if normal individuals are born with 2 good copies of this DNA mismatch repair gene and individuals with Lynch syndrome inherit 1 damaged copy, what was happening at about 15% of colorectal cancer was that the genes were perfectly good, but the DNA promoters for the genes were being silenced by a chemical modification, the acquisition of methylation of the DNA in the region of the MLH1 gene. We went on to show that this was a general mechanism that could give rise to cancers. We studied families with hereditary diffuse gastric cancer. This is a condition that is caused by inheritance of a damaged copy of the gene encoding for E-cadherin. Here you see a stain of the stomach of an individual that has inherited this condition. The stain, the brown stain, shows the normal E-cadherin and the normal gastric pits of this individual. And here you see this diffuse gastric tumor that is missing E-cadherin. Here are two normal crypts that remain in the tumor. And when we asked what the second step was that gave rise to these cancers, again, we found in many of these individuals, the second copy of the gene for E-cadherin was perfectly normal, but again, it had been damaged by the acquisition of methylation of the DNA that was silencing the gene. So we wanted to understand what the basis of methylation was in these GI malignancies. And we took a collection of 21 colon cancer cell lines. We treated them with the DNA demethylating agent 5-azacitidine, which is now used as a drug to treat leukemias. And we looked at all of the genes that were activated by stripping the methylation from the DNA. And we asked whether those genes were directly methylated. And that led us to an unexpected finding, which was a gene known as vimentin had a cluster of residues right at the beginning of the gene that in human colorectal cancers were selected to again acquire this methylated modification. We could detect that methylation with a simple assay. And much to our surprise, we found this modification was present in 80% of colorectal cancers. So this was the most common modification of DNA in colorectal cancers. And in a translational jump, we showed that this modification was so robust that we could detect it even in DNA that was isolated from human stools. So here were the stool DNAs from a group of normal individuals that were negative in this test. And here were DNAs from a group of patients with colorectal cancer, where we could easily detect this cancer-specific methylation. And we found that this methylation was present early. We could use it to detect over 80% of individuals with early stage 1 or 2 colorectal cancers. This became a commercial test. We developed this in collaboration with Exact Sciences in the United States, and the test was brought forward as ColoSure, marketed through LabCorp. And it was the direct predecessor of what is now known as Cologuard. This was the ColoSure advertisement. And it was the predecessor of a test now known as Cologuard, which is used for non-colonoscopic, non-invasive screening for colorectal cancer. But of course, colorectal cancer can easily be screened for by colonoscopy, and that's still the most robust method for screening for it. But there are many places in the GI tract where endoscopy is not standardly used. And so we thought we should take our vimentin marker forward and look at other regions of the GI tract. And in particular, in the US at least, esophageal cancer has been a remarkably increasing problem. Here you see the increase in the incidence of esophageal adenocarcinoma over the last 3 decades in the US. It's a miserable disease. The 5-year survival is less than 20%. The death toll from ovarian cancer in the States now exceeds that from ovarian cancer, and it's seldom detected early. But there is an opportunity to intercept the development of esophageal adenocarcinoma, and that's because the cancers have a precursor lesion, which is shown here, which is called Barrett's esophagus. It's the transformation of the normal squamous esophagus to this columnar epithelium. Now, we know that this is often a response to chronic reflux disease, chronic esophagitis. And we know that there is a stepwise progression from the Barrett's to Barrett's with dysplasia before the cancers develop. So there really is an opportunity to detect the Barrett's and the dysplastic Barrett's as a way of intercepting the cancers. And indeed, dysplastic Barrett's can be eradicated endoscopically. So the question is, can we detect the Barrett's and the dysplasia and thereby prevent the cancers? Now, there is an opportunity to do this. For one thing, the Barrett's arises in a stereotypical location at the GE junction or just above it. Now, the problem is that Barrett's is a neoplasia, but it doesn't produce any detectable circulating tumor DNA. Even early stage esophageal adenocarcinoma does not to produce much circulating tumor DNA, and yet it's already an incurable lesion. So blood tests are not the answer. And endoscopic screening is not a good answer because it's only been recommended for those with chronic reflux disease with multiple risk factors. It requires a day off of work because of the sedation. And at least in the States, it simply hasn't been well accepted by either primary care physicians or patients as a means for Barrett screening. And the result of that is that over 90% of the Barrett's esophagus in the States goes undetected. And 95% of people who develop esophageal cancer never knew they had Barrett's. So we saw biomarker based detection as a way to change this, to approach this problem in a new way. And we realized there are several challenges in approaching the disease this way. We need biomarkers that can detect Barrett's. We need a device that can replace the endoscope. And we need biomarkers that can tell us when the Barrett's will become dysplastic. So let me show you the three solutions to those challenges. This is the team that worked on the problem with us. Now, the first step was an observation that we brought forward in cancer epidemiology and biomarkers, which was simply that the vimentin marker that we had found in the colon was highly present in Barrett's esophagus. In fact, methylated vimentin is an even better marker of Barrett's than it is of colon cancer, as over 90% of the Barrett's has it. We were able to detect it initially using cytology brushes that were passed through an endoscope into the esophagus. And here were the normal individuals. Here was the high levels of methylation in individuals with Barrett's or dysplasia or cancer. And we did a clinical study of over 300 individuals and showed that we could divide this into a training set and a validation set. They both looked absolutely identical, with over 90% sensitivity for detecting the Barrett's at over 90% specificity, if the individual had more than 1% of methylated vimentin. We went on to do an agnostic genome-wide screen for additional methylated loci that could mark Barrett's. We found the CCNA1 promoter was a second vimentin-like gene. And we could combine those into a panel. They were both very, very similar. And when we put them together, we could detect with over 90% specificity, over 90% of the Barrett's or the dysplasias or the cancer. It was highly specific for Barrett's in a histology study. We looked at other diseases of the esophagus, including chronic carditis and eosinophilic esophagitis. They were all negative for vimentin. So the challenge now became, how do we replace the endoscope? How do we do something that's simple for patients that doesn't require sedation? And we came up with the idea of a swallowable balloon. And this is the device that we developed and engineered. It's the size of a vitamin pill or a dime in the US. It has a little capsule that can be swallowed. The capsule is on a silicone catheter, and inside the capsule is this textured balloon. So the capsule is swallowed, it goes into the stomach, and then the balloon is deployed and just pulled back through the gastroesophageal junction to basically do a touch prep of the GE junction. And then the air is pulled out, and the balloon pops back into the capsule, which protects your sample now from both dilution and from contamination. So this is, in a sense, a molecular pap smear of the esophagus. This device has now been commercialized and FDA approved. In the US, it's called Esoc. It's also gotten EU approval. And our methylated DNA panel has also been brought forward commercially. It's called EsoGuard, and it has breakthrough device designation in the US and approval in the EU. So the technology is moving forward into patients. The advantages of the balloon is that it's a small in size, it's easy to swallow, it's easy to remove without any patient discomfort. because it protects the sample, it minimizes contamination, and maximizes signal. Oops, let's go forward. And this is just a picture that shows how the balloon works in picking up this touch prep from the esophagus. And it's easy on the patient to deploy. This is my colleague, Dr. Amitabh, who's head of our endoscopy team. Here you can see he's testing the balloon. And there it is in its capsule. And this is one of our actually GI surgeons easily swallowing it down. And there it is easily being retrieved. And then after the balloon's retrieved from the patient, it can just be cut from the capsule and sent to the lab for assay. So we did a clinical trial with 156 patients. Over 80% could swallow the balloon. We took 86 of these individuals who had not had any prior esophageal procedures. Oops, let's go back one. Okay. We excluded individuals that had prior ablation. We found that on average, the procedure took just over 3 minutes. 93% of the individuals said they preferred the procedure to endoscopy. We'll be happy to repeat it again. And the scores for discomfort or anxiety were very low on our questionnaires. So when we took the balloon device and added our methylated DNA panel, we were very gratified to see that the balloon was actually every bit as good as were directed cytology brushings. The AUCs, the areas under the curve for detecting Barrett's, were very high for both our biomarkers. And in conjunction, we had 92% specificity and 90% sensitivity for detecting Barrett's. And we published this in Science Translational Medicine a little bit over 36 months ago. And this was this very nice graphic that the Science Translational Medicine Journal put together, explaining the advantages of biomarker-driven detection for Barrett's versus traditional endoscopy. And this was highlighted by the National Cancer Institute in 2018 in their report to the US Congress as one of the important advances of the year. So I mentioned the technology has been licensed to a company, Lucid Diagnostics, which is now making it commercially and clinically available for patients to use. In 2021, we've seen FDA approval and CE mark approval, as well as recognition of the technology, winning a Silver Edison Award for innovation. So we have ongoing clinical trials now of the technology nationwide, some sponsored by the NCI, some sponsored by Lucid, the company that's licensed the technology. And these are initially aimed at a screening cohort of males over 50 who have a history of chronic heartburn and any additional risk factor, which can be either obesity or Caucasian ethnicity or a history of smoking. However, detecting Barrett's is only the first step toward stopping deaths from esophageal cancer. The procedure, at least in the US, for individuals who have known Barrett's, is to be followed with endoscopy every 3 years with random biopsies during those endoscopies in the hope of catching dysplasia before it progresses to cancer, and then eradicating the dysplasia by burning it out endoscopically. And this is a highly imprecise method of monitoring Barrett's. The cancers that are detected by random biopsies are often detected late. 40% of them are already metastatic at the time they're detected, even in individuals who are under surveillance. So we need a better method of detecting progression for Barrett's. The problem, of course, is that you're looking for the molecular needle in the haystack, in that you have the esophagus, it's been changed into Barrett's, and you're looking for these 1 or 2 clones where there's dysplasia. And so the question is, how do you do that? So to approach that problem, we considered that this really is a very analogous problem to the problem of developing a blood test for cancer. The challenge of developing a blood test for cancer is to find a rare cancer DNA and a lot of DNA that's in blood. We're asking to find a rare progressed DNA in esophagus that has a lot of DNA that's quite normal. And so we teamed up with our friends and colleagues in the Vogelstein lab at Johns Hopkins who were working on blood tests for cancer. And we adapted their technology for blood to instead work on brushings from the esophagus. And this is a schematic of what we came up with. It's a technology we call BAD. We published it just a few months ago in Gastroenterology. And the idea is that individuals who are known to have Barrett's and are going to have an endoscopy surveillance will have a brush of the entire Barrett segment, not random biopsies, but a brush that gets all of the Barrett cells. We extract the DNA from those cells, and then we look for very subtle changes, just as if this were a blood sample, in the distribution of DNAs on the different chromosomes that would indicate that chromosomes have been gained or lost somewhere in that Barrett segment. So the first approach was just to ask, do you see imbalances of chromosomes? This is, of course, called aneuploidy. And this is the distribution of chromosome balance in the normal esophagus. And here you can see that in the cancers, individuals with cancers, they're very unbalanced in their chromosomes. They have a lot of aneuploidy. Individuals with high-grade dysplasia also are very unbalanced. And as are most individuals with low-grade dysplasia, although some low-grade dysplasias are really quite normal and are probably an overcall by the pathologist. The surprise was that when we looked at the Barrett's, it falls into two groups. A lot of Barrett's still has normal chromosomes, but a third of Barrett's is in fact already not balanced, not perfectly balanced for chromosomes. So that's a problem because we can't say that a third of Barrett's needs to be burned out. That's way too many patients. So we took a look at exactly what chromosomes were being gained or lost in the cancers. And we found there were just a few chromosomes, there were 12 chromosomes that were very recurrently targeted for imbalance in esophageal cancer. And we didn't need all 12. We could actually capture all the information in this whole panel with just the 6 chromosomes shown in red, 8q, 1q, 20q. I won't read them all off, but the idea is a small panel of 6 chromosomes was enough to recognize when the Barrett's was actually dysplastic. Oh, here's the list here, 1q, 9p, 12p, 17p, 20q. So we developed an algorithm. We first said, are chromosomes imbalanced? If they're not, we call that not bad. If we see evidence of chromosome imbalance, we have to look further, and we look at the specific list. And if the specific chromosomes on this list aren't there, then we're okay again. We call it maybe bad. But if there's chromosome imbalance and it involves any of these 6 driver chromosomes that are seen in cancer, that we called very bad. So this was our training set, and you can see here is the number of samples that are aneuploid, that are chromosome imbalances. Most of the cancers do, the great majority of the dysplasias do, but here was the problem, the third of Barrett's. If instead we use our BAD approach and ask if there are only specific driver chromosomes that are imbalanced, we can screen out most of these Barrett's that are actually not problematic and focus just on these small numbers that have the imbalances that are seen in cancer. Now, this was a training set. 90% of the cancers were very bad. 60% of the high-grade dysplasias were very bad. Only 5% of the Barrett's was very bad. We needed a validation set, and we took another group of patients. We again saw the same behavior. Many of the Barrett's had overall chromosome imbalances, but only very few had imbalances of these driver chromosomes seen in malignancy. And our very bad approach could detect 96% of the cancers, nearly 70% of the high-grade dysplasias, and only 7% of the Barrett's. So we actually did better in the validation set than we had in the training set, which was quite surprising to us, but suggested the technology was robust. We missed a few high-grade dysplasias, but the ones we missed were focal in some cases, meaning they were less than 1 millimeter across. So we would expect they would be picked up with a repeat exam a few months later. And most of the misses, though, were in either very short or very long Barrett's. And we expect in the very long Barrett's, we simply hadn't brushed robustly enough. And now we've moved to a bigger brush to make sure we capture all of the Barrett's that's there. So can this supplement or replace our random biopsies? Well, one question is, can it predict which patients are going to get into trouble? And we followed the cohort that we had studied for the next 36 months. We had 4 patients with normal just Barrett's, non-dysplastic Barrett's, who we had called very bad. And over those next 36 months, 2 of those 4 progressed to cancer. None of the Barrett's that we had called not bad or maybe bad progressed. So we believe that the patients in whom that the bad algorithm detects very bad are at very high risk for progression, even if they don't have high-grade dysplasia yet. And we're now just about to launch a clinical trial looking at individuals with low-grade dysplasia to see if we can distinguish individuals with low-grade dysplasia who are truly at high risk for progression and need ablation versus those who are not very bad and probably have been overcalled and just have reactive changes. We're also about to launch a study of the patients who have non-dysplastic Barrett's to try to confirm that this small group of non-dysplastic Barrett's we call very bad are again the individuals that are at high risk for cancer. So what we have found is that methylated DNA is a molecular biomarker that can detect esophageal precancers of Barrett's, that this engineered sampling device, our swallowable device, can allow us to dispense with the endoscope for the screening, and that once we have individuals with Barrett's who need endoscopy to follow them, that replacing random biopsies with a brush and a DNA test can allow us to molecularly detect the individuals who have small foci of dysplasia, hopefully more sensitively than random biopsies will, and also allow us to identify individuals who are at high risk for progression, even who do not yet have biopsies with dysplasia. I want to thank our clinical collaborators at Hopkins and at UNC and at Mayo and the Cleveland Clinic who have helped us on these clinical trials, as well as acknowledging our funding from the NIH and for our colleagues at Hopkins. And with that, I thank everybody for their interest and attention this evening. And I can stop sharing my screen and turn the podium back to Dr. Abu-Hamor.
Thank you, Professor Marquez. This is an excellent and outstanding presentation. Definitely we're pleased to see somebody like you and with your caliber interested in this cancer. We believe that probably esophagus cancer is the 8th most common cancer worldwide, and it's the 6th deadliest cancer. And as you said, waiting for alarming symptom is really too late. These patients are diagnosed at late stage, tumor is aggressive, very poor survival. Now, we'll open the floor for questions. We can take questions and then we'll go through them. We are waiting for that. But while we are waiting, in UAE, we did a few years ago a retrospective study in my center and some centers in Sharjah and Dubai with my colleagues, and we found that the prevalence of Barrett's probably 0.4% in patients who get upper GI endoscopy. So probably we don't know that this will go up because obesity is also coming to our area, and obesity, smoking, maybe with the chronic GERD is a high-risk patient. Do you pick targeted patients you do the balloon for? Because, you know, GERD is chronic, And sometimes just by clinical symptoms, you cannot distinguish who has Barrett, who have Barrett's and versus who does not or don't have Barrett's. So what do you do in this case? Do you, about selecting patients for screening with the balloon? Please.
So there are many interesting insights in what you've just said, Dr. Abu Hamor. The first, of course, is, as you mentioned, that Barrett's is very rare in the UAE, which is a good thing. I'm pleased to hear it. As you and I were chatting, when I was in medical school, I never saw a case of either Barrett's or a case of esophageal adenocarcinoma. All of the esophageal cancers were squamous, and they were all in the upper esophagus. But as I showed on that graph, esophageal adenocarcinoma has been the most rapidly increasing tumor and cause of cancer death in the States over the last 30 years. And my guess is that sadly, similar to colon cancer, we're likely to see this epidemiology spread to the rest of the world. So in the same way that colorectal cancer was once quite rare outside of North America, it's now become quite common in many places. Southeast Asia is a good example where it was once quite rare. And I fear that the same will be true of Barrett's and of esophageal adenocarcinoma. So I'm hoping that we will be able to pilot this technology for easily managing it before it becomes a cause of mortality in other portions of the world. In terms of I'm sorry, go ahead.
No, go ahead, please.
Okay. In terms of managing Barrett's and how to use the balloon, we're still working out what the indications should be. Right now, our initial studies basically was a population of Caucasian males over 50 with GERD, with chronic reflux. That's a group that has between 5 and 10% incidence of Barrett's. And our positive predictive value in our simulations is between 30 and 40% if we use the balloon and the biomarkers to test that population, which would be about 20 million individuals in the States. We would like to expand the approach because 40% of esophageal adenocarcinoma in the US right now arises in individuals with silent reflux. They're stoic and they don't complain of heartburn. And our epidemiology suggests that we can identify many of those individuals if we simply take folks over 50 with 2 clinical risk factors. And risk factors can be male gender and obesity, for example. So we believe that at least in the US, where obesity has become unfortunately all too common, taking Caucasian males over 50 will again define a population with about a 5% incidence of Barrett's. And we're just getting ready to launch clinical trials that would study that population with the balloon technology and not require individuals to report symptomatic GERD. So I think that's where we see the technology evolving. And if we can validate the technology in individuals that not only have symptomatic reflux, but silent reflux, then I think we really will have a way to prevent the Barrett's from becoming a source of silent cancer. Because as you pointed out, by the time individuals have dysphagia, they're basically have a death sentence.
Thank you for your clear, nice answer. We have a few questions. I will forward it to you. We have a question that says, what is the patient preparation before Esoc balloon? Is there any specific instructions to be given? Patients should come NPO or stop PPIs or anything?
No, we haven't found that anything is necessary. In practice, because our studies were being done in individuals coming in for endoscopy, they were NPO overnight. But we've done studies in the lab. Actually, our good colleague Amitabh Chak, who you saw administering the balloon, was also the first person to swallow it, and he's tried swallowing it, drinking water, swallowing it with eating bread. We haven't found anything that really affects the performance of the balloon.
Thank you. There is also one from one of our colleagues, two questions in one. Did you compare it with cytosponge? And the second question, do you have any studies about Barrett's microbiome signature?
Two very good questions. So we would love to compare it to cytosponge. We couldn't do those studies because cytosponge isn't available in the States. There were some studies of cytosponge in the US. Nick Shaheen, who writes the guidelines for the American Gastroenterological Association, led them. In the States, cytosponge underperformed the reported performance of it in the UK. And there was a problem with a detachment of the sponge during the studies. But I think probably the greatest advantage, well, there's two advantages of the approach comparison to cytosponge, we think. One is cytosponge right now is an immunohistochemical assay for TFF3, meaning there is some subjectivity in the interpretation. And that subjectivity grows if it's not being interpreted in a single reference lab. So we like the idea of using a DNA test, which is an automated test that can be automated and done at scale with a standard cutoff compared to a qualitative test. We also think that patients will find the balloon more comfortable. Cytosponge, of course, expands, and it's that expanded sponge that's pulled through the throat to retrieve it, whereas the balloon can be constricted back down to the size of the pill, the capsule, for more comfortable extraction.
We have another question, probably a technical question. You mentioned clearly that this device is FDA approved, and the question is, the Esoc balloon available in other countries, up to your knowledge, Professor?
So Lucid is just scaling up the manufacture of it. It has a CE mark approval. So I would anticipate that the intention is to make it available in the EU, but I actually don't know whether that commercial availability has begun yet. I know that there are clinical trials in the EU that are ongoing, but whether it's available outside of clinical trials in the EU, I'm not sure. That's a good question. I think I also skipped over the question about the microbiome. The microbiome is a very interesting topic for research. And I think that that actually the balloon would be a nice way of getting samples that could be used for microbiome research.
Another question, do you think this balloon device is more rigid compared to the Esoc cap? And they say it's thinner, flexible, and being retrieved easily. Do you have comparison between the two? And is it truly that this Esoc is really rigid?
No, Esoc not rigid. Esoc is a soft balloon that it's inside of a capsule, which is the size of a vitamin pill. The capsule wall is rigid, but small. But once the capsule goes into the stomach, the balloon is deployed to provide a touch prep of the GE junction. And that's soft, that's squishy. So when it comes through the GE junction, there's a little feeling of pressure, but the balloon slips through the junction, and then it's deflated. So it goes back into the capsule. So after the touch prep, the balloon is gone. It's totally deflated back into the capsule. So it's not rigid. The balloon's not rigid at all.
So patient tolerance is very good. As you mentioned in one of your slides, the majority will do it again if it's necessary or needed. So I believe it is comfortable. We have another question, says, with the increase in the prevalence of esophageal cancer in Barrett's esophagus, are we expecting change in paradigm modification, the screening guidelines in the near future? And
well,
what do you suggest for the guidelines for screening?
We want to see the standard of care change for this, for screening for Barrett's. There's no medical reason that we shouldn't be screening for Barrett's the same way we screen for colon adenomas. The difference is simply that colon adenomas are present, in a third of the population over 50 in the States, whereas Barrett's is present in 5% of the population, at least of obese white males over 50. So the cost benefit analyses for endoscopic screening have not been as supportive of esophageal screening as they have been for colon screening. But if you replace endoscopy with a simple outpatient swallowable balloon, that analysis is turned on its head. So we absolutely want to see an indication for screening for Barrett's for males over 50. And we are working toward the clinical trials that would validate that approach.
Thank you. There's another technical question, actually. From the balloon collection, when you send it to the lab, is it really biopsy or is it cytology, what you request in the lab?
No, it's a DNA test. All right, so the cells on the balloon could, of course, be used for cytology, but cytology is hard in the esophagus. So we extract the DNA from those cells, and then we sequence it looking for that methylation modification. So it's a DNA test like any DNA test.
I think we have some more questions, but they are overlapping with previous questions that they were asked to the professor, so I will not go through them. And with that, I would like to thank you, Professor Marquez, for this interesting presentation and sharing with us something new that really will have an impact on clinical practice. As we discussed in chat before, that esophagus is very deadly, and probably most people in literature agree that it is one of the least cancers that has work at, and scientists are looking at it, but it's not as much as you expect for such a cancer with a rising prevalence. Most of us know that the squamous cell carcinoma surpassed adenocarcinoma. Maybe adeno was rare before the late 70s. And from the 80s till now, we see that the squamous cell carcinoma plateauing in some countries and declining actually in Western and USA as well, and with a sharp rise in the adenocarcinoma of the esophagus. So probably we're gonna see more and more, and probably esophageal cancer maybe will move to be to the fourth or third common cancer as adenocarcinoma. What do you think? And we'll close with that, Professor.
I expect that the epidemiology that we've seen in North America and in Europe will unfortunately spread to the rest of the world. We don't know what's driving the shift from squamous to adeno in the esophagus, but it has certainly increased steadily over the last 3 decades in North America and Western Europe. And the history of colorectal cancer has been, we see this epidemiology first in North America and in Europe, and then it spreads. And I fear that's what we will see again. So I hope that this is a technology that will allow us, at least if Barrett's is going to spread, to prevent the cancer from spreading.
Well, thank you so much, Professor Marquez. That's an excellent. I would like also to thank the Hamdan Award trustees members for their tremendous work, and the committee who worked to put this together tonight, and also our colleagues who attended this conference. Hopefully that this will motivate us, or at least some of us, to look at this esophagus cancer and pick up on their research on this issue. Thank you so much, and have a lovely evening for people with the afternoon time, and lovely morning like our professor in Ohio, Cleveland.
A great pleasure to see you again, Dr. Abu Hamor, and I hope soon we will all be able to see one another in person, inshallah.
Inshallah, looking forward. Well, thank you, everyone.